Do you have a lot of names?
Like I have like this is something easier because like I did
Yeah.
like so what I did. Yeah, and then like some people messed it up. So I just went like one and then I also make my last name just add
People can prepare for I'm working with my PhD. Her name is Liza. She's called herself or friend. Immediately when it comes to Canada.
Okay, so we're going to start. I have the distinct pleasure to introduce Luka Milosevic. He's going to talk about neurophysiological mechanisms of deep brain stimulation. Just a couple of things about Luka. Luka did his undergraduate at Ryerson, and then he joined us at UFT. He had an exceptional mentor, which is Bill Hutchinson. He had another guy, I don't know who he is. Anyways, he finished his PhD really well, and now very good academic. He's now at the University Tübingen. It's a very well-known center in Europe, especially in Germany, for neurology and neurosurgery. And he's part of this university Tübingen. And these things work together seamlessly. So, delighted to have you with us. And
yes, thank you very much for the introduction. Thanks for having me here to talk. So I'm currently doing a postdoctoral fellowship at the University of Tübingen as a corporate.
can you hear him? Sorry. No. Okay.
So as Dr. Popovich mentioned, I'm doing a postdoctoral fellowship at the University of Tübingen in Germany. I'm with the Center of Integrated Neurosciences and the Department of Neurosurgery, and I have the pleasure of working with these very ambitious clinical team at the University of Tübingen. Part of the contents of my presentation today, I won't use this, I'll just speak about it, pertain to the research that I did during my PhD studies here at the University of Toronto, where I was fortunate enough to work with Bill Hutchinson and neurosurgical fellows and neurologists at Toronto Western Hospital. So for those of you who perhaps are not familiar with deep brain stimulation, or DBS, it involves a surgical implantation of chronic indwelling electrodes into a specific target region of the brain. The electrodes are programmed externally via a pacemaker that's implanted in the chest, and they're typically used to deliver continuous high-frequency stimulation to a particular target region of the brain. The target region can vary depending on the disease or the indication, but generally stimulation is delivered to a very specific, deep subcortical structure that's implicated in the pathology of said disease. The most common indication for DBS is Parkinson's disease, but it's also often used to treat other movement disorders, including essential tremor and dystonia. So a typical DBS procedure involves several steps, and you have to keep in mind that patients are awake throughout the entire procedure because their feedback is imperative throughout the procedure, and also allows us to do some interesting research. So firstly, CT and MRI images are used to essentially plan where the location of the electrodes will go, and that's determined based on essentially triangulating the target structure with respect to a frame that's attached to the patient's head. It's called a stereotactic frame. And then under local anesthesia, an incision is made and two small burr holes are drilled. And essentially we use an apparatus, a drive, which drives the electrode to its final target location. But prior to implanting the final DBS lead, we perform microelectrode recordings. And these microelectrode recordings are used for electrophysiological mapping. So they're used to confirm the target location, but we also use this as a means to do research. So at Toronto Western Hospital, we use a unique microelectrode recording setup. So during each surgery, we advance 2 very closely spaced microelectrodes. And each microelectrode is used to record action potential firing, which is the electrophysiological signal acquired from one signal neuron within the brain. We can also use either of these electrodes to deliver electrical stimulation. The typical electrophysiological mapping procedure involves delineation of the different subcortical structures along the surgical trajectory based on their characteristic firing rates and firing patterns. So essentially each of these structures speaks its own language, and we get an idea of where we are within the brain based on how these neurons behave. In the case of Parkinson's disease, what we're looking for is a small structure called the subthalamic nucleus. It's a structure of the brain that's within the basal ganglia. And the basal ganglia is a collection of subcortical structures which is involved in the volitional drive of movements. So essentially, as a collective, these structures help to decide whether voluntary movement should be facilitated or whether voluntary movement should be suppressed. And in Parkinson's disease in particular, the subthalamic nucleus is known to be overactive, and essentially that leads to an oversuppression of movements. And subsequently, it's been implicated as the surgical target region for surgeries for Parkinson's disease. So at the top of a surgical trajectory targeting the subthalamic nucleus, we may encounter slow bursting reticular thalamic neurons. As we advance the microelectrodes, we come near our target of interest, which is the subthalamic nucleus, which would have irregular firing patterns and firing rates of around 20 to 60 hertz. And below here is the substantia nigra pars reticulata, which has fast, regular neuronal firing. If, for example, we don't encounter any neuronal activity along the trajectory, we can then stimulate. So, for example, if we stimulate and the patient experiences a motor contraction, we know that we're activating cortical spinal tract fibers of the internal capsule, and we know to move our surgical trajectory more posteriorly. Or if the patient experiences paresthesias or tingling, we know that we're activating sensory fibers of the medial lemniscus, so we'd move more anteriorly. But once we're happy with the electrophysiological localization of the subthalamic nucleus, that's when the DBS electrode is implanted, which is the one that stays in the brain permanently. So if we're in the right spot, subthalamic deep brain stimulation has a remarkable therapeutic efficacy for the motor symptoms of Parkinson's disease. So you can see that this patient here has very labored hand movements, but post-operatively the stimulation on, her movements are fast and fluid. Here you can see that she has a large degree of difficulties with her gait. She can't walk without cane, and in particular she has difficulties with turning around. The so-called freezing of gait is a very, very debilitating feature of Parkinson's disease. But here she is again with stimulation on. So it's pretty impressive. There we go. So in Parkinson's disease, we most commonly implant electrodes into the subthalamic nucleus, which I've spoken about. We also sometimes put electrodes in the globus pallidus, which is another structure within the basal ganglia. There are some indications that combined STN and SNR, substantia nigra pars reticulata stimulation, can be very helpful for resistant gait and postural symptoms of Parkinson's disease. And implantation of electrodes into the ventral intermediate nucleus, which is a substructure of the thalamus, is commonly done for the treatment of essential tremor or tremor related to Parkinson's disease. I've already demonstrated sort of the electrophysiological mapping procedure for STN DBS, where we map for the STN and the SNR, but we also do electrophysiological mapping for the GPI as well as the VIM. But in the presentation today, I'll talk about some of my research which pertains to the structures that you see here in bold. So we know that DBS is a remarkably efficacious therapy, and we know where and how to implant electrodes. However, the exact mechanisms of action of electrical stimulation of these subcortical target structures remains unknown. main objective of my research was to try and figure out what exactly is going on when we deliver electrical stimulation to these particular brain regions. And in particular, I wanted to investigate how neurons, individual neurons, within various subcortical DBS target structures respond to electrical stimulation at different settings. And basically we were able to shed light on three main mechanisms of electrical stimulation, which are synaptic modulation or synaptic activation, synaptic depression or synaptic fatigue, and increased inhibitory synaptic plasticity. And we were able to do this because of that unique microelectrode recording setup that I mentioned previously, where we're able to record from individual neurons, and with the immediately adjacent electrode, we can deliver electrical stimulation. So the first mechanism is synaptic modulation. So it's very well known that clinically therapeutic DBS is delivered at a high frequency. Typically, so the patients are implanted with the electrode, the stimulator is programmed usually to 130 hertz, and that's considered high frequency stimulation. So low frequency stimulation don't have therapeutic benefit. So what we were interested in doing was determining the frequency dependent effects of electrical stimulation on single neurons within the DBS target structure in Parkinson's disease, which is the subthalamic nucleus. We also did the same protocol in the SNR as well. So we delivered trains of stimulation at 1 hertz, 2 hertz, 3 hertz, 5 hertz, 10 hertz, 20 hertz, 30 hertz, 50 hertz, and 100 hertz. We did this in 22 patients in several STN and SNR neurons. And one of the things that we looked at was the neuronal firing rate during electrical stimulation after removal of the very narrow stimulation artifact. Essentially, we took the value of how quickly the neuron was firing during stimulation and compared that to its baseline firing rate before any stimulation was delivered. And what we found was that as we increased the stimulation frequency, the firing rates were progressively reduced in both structures. So as we approached therapeutically beneficial stimulation frequencies, the neurons became more and more inhibited. And from these curves, you can see that the SNR, the substantia nigra pars reticulata, was more sensitive to this stimulation than the subthalamic nucleus. So the SNR was able to be completely silenced at 50 hertz stimulation, while the STN required 100 hertz for neuronal silencing. So this is a schematic of a neuronal cell body. You should imagine that the little red dots you see are each inputs that this neuron is receiving from other structures within the brain to which it's connected to. And each of these little connections is called a synapse. So when we deliver electrical stimulation, we're simultaneously activating all of these presynaptic terminals, causing release of neurotransmitters from these terminals, which are the chemical compounds that neurons use to facilitate or inhibit communication with one another. So the SCN and the SNR both have predominantly inhibitory or GABAergic synapses located on their somas, which, when activated, reduce the likelihood of these structures generating action potentials. So this may explain why these neurons became more and more inhibited as we increase the stimulation frequency. Furthermore, histological studies have shown us that the SNR has 90% GABAergic synapses on the soma compared to 60% which are in the STN, which may explain why the SNR was more sensitive to the stimulation than the STN was. So it appears that an important mechanism of therapeutic high-frequency stimulation is neuronal inhibition. So we conclude that electrical stimulation causes release of neurotransmitters from the presynaptic terminals, but it appears that the net effect is dependent on the weighted composition of inhibitory and excitatory synapses present on the soma. So it might seem a little counterintuitive because I'm saying it's synaptic activation. So you might think that a synaptic activation would cause firing of action potentials, but since we're activating inhibitory terminals, what it's doing is decreasing the likelihood of firing action potentials. And since the STN and SNR both have mostly GABAergic synapses, the net effect is inhibitory. So one might assume then that the neurons becoming completely silenced at high stimulation frequencies may be the result of a temporal summation of inhibitory responses. So if one stimulation pulse causes the neuron to be inhibited for a certain amount of time, if you deliver the stimulation pulses fast enough, that neuron can be inhibited indefinitely. But it turns out that's not exactly what was going on. So we were able to debunk this theory by stimulating a structure with predominantly glutamatergic synapses, which are excitatory synapses. So as we would expect, each stimulation pulse caused that neuron to fire several action potentials instead of being inhibited. So due to the activation of the predominantly excitatory synapses present in the soma. And at 5 Hz stimulation, which you see here, this neuron entrained its firing to the stimulation pulses. However, when we stimulated at a high frequency of 100 Hz, the neuron was also excited for a very brief period of time at the start of stimulation, then it also became inhibited. So our temporal summation theory would suggest that this neuron would be excited indefinitely. However, we can see here that that was not the case. Which brings us to our next mechanism, which is synaptic depression, or synaptic fatigue. So in the SNR, the substantia nigra pars reticulata, which is a structure we record from very often, we were able to nicely study this phenomenon because in this structure, each stimulation pulse produces a pronounced evoked field potential. And this is an inhibitory evoked field potential, which I might refer to as an FEP presentation. This FEP is most likely the result of the very high predominance of inhibitory inputs that the SNR receives. So as I mentioned, when a stimulation pulse is delivered, we're activating the presynaptic terminals. This results in the release of neurotransmitters, which is our synaptic activation mechanism. When GABA is released, it binds to GABA receptors, causes an influx of chloride ions, which are negatively charged ions. So we measure a positive going voltage peak deflection because we're measuring extracellularly. So you can imagine that each of these evoked potentials is kind of like a measure of how much neurotransmitter is being released. And as I mentioned in the SNR, each pulse evokes one of these potentials. So we were able to show the phenomenon of synaptic depression very nicely by studying the behavior of these evoked potentials during stimulation at various stimulation frequencies between the consecutive stimulation pulses. At low frequencies, we record robust, constant amplitude evoked potentials that don't change with consecutive stimulation pulses. So we call this our baseline amplitude. But as we increase the stimulation frequency, we found that the first FEP, the first evoked potential in the 30 Hz stimulation train, was actually larger than the baseline amplitude. So this is kind of an interesting observation, but I'll come back to this later on. It doesn't have anything to do with synaptic depression. For now, what we're interested in is that the last evoked potential in that 30 Hz stimulation train, it was significantly smaller than the baseline amplitude of evoked potentials at lower frequencies. And that's because there's a rapid attenuation of successive evoked potentials throughout the stimulation train. And at 50 Hz, you can see an even more rapid attenuation, and this effect is indicative of synaptic depression or synaptic fatigue. So if each FEP is a measure of how much GABA is being released, you can see that at high frequencies, less and less is being released with each consecutive stimulation pulse, and the faster the stimulation pulses are delivered, the more rapidly these evoked potentials are depressed. So we conclude that synaptic depression via neurotransmitter depletion occurs during high-frequency stimulation, since repetitive stimulation at high frequencies does not allow synapses enough time to recover and release their neurotransmitters again. And this effectively causes a functional deafferentation or a functional lesion within the structure you're stimulating continuously at a high frequency. And this likely explains why this neuron here was also inhibited rather than being excited indefinitely. So we want to investigate how this mechanism may have direct clinical implications. In order to do this, we delivered therapeutically relevant stimulation trains at 100 Hz and 200 Hz in the ventral intermediate nucleus, which is a thalamic substructure, and it's also the DBS target structure for patients with essential tremor and tremor-related Parkinson's disease. And since I mentioned that these patients are awake during surgeries, we're able to simultaneously record action potential firing while measuring their tremor intraoperatively using an accelerometer. And just one quick important thing to note is that the ventral intermediate nucleus has a predominance of glutamatergic synapses as opposed to inhibitory GABAergic synapses. So I've already mentioned that the release of GABA hyperpolarizes the neurons and inhibits them, whereas glutamate release would result in depolarization due to an influx of sodium ions. With enough depolarization, you would expect firing of several action potentials. And I showed that figure previously where each stimulation pulse resulted in firing of action potentials. And indeed, what we found was that there was a transient stimulation-induced burst of neuronal activity at the start of the stimulation trains, and this was quickly followed by a successive suppression of neuronal activity. But the interesting thing was that both of these phenomena, the initial burst and the subsequent inhibition of neuronal activity, had behavioral correlates. So we found that the transient stimulation-induced burst at the start of the train led to a very brief, short latency muscle contraction and tremor phase reset. So we'd get the patient to hold out a water bottle, their tremor would kick in, and we're measuring essentially the tremor with an accelerometer, and right as we initiated the stimulation, there would be a quick muscle contraction, it would change the phase of the tremor, and then depending on whether the neuron was inhibited or not, something might happen to the tremor. I'll get to that in a second. But essentially, we conclude that most likely this transient driving eventually relays down the spinal cord, causes a muscle contraction. However, after this initial burst, we found that when the stimulation was insufficient in suppressing the neuronal firing, the tremor also persisted. And this commonly happened at 100 Hz stimulation, whereas after the initial burst, if the neuron was inhibited entirely, the tremor was also suppressed. And indeed, we found that the amount of neuronal inhibition at each frequency was associated with the amount of tremor production. So the more the neurons were inhibited, after that initial burst, the more the tremor was suppressed. So to summarize these findings, initially glutamate release, which is the excitatory neurotransmitter, causes a transient excitation of neuronal activity, which leads to a brief muscle contraction. But shortly after that initial driving, the neuronal inhibition kicks in, most likely due to synaptic depression, which seems to be necessary for tremor suppression. And indeed, neuronal inhibition appears to be an imperative mechanism, therapeutic mechanism of action of DBS. So the final mechanism that I'll talk about is what I promised I'd come back to, which is enhancement of inhibitory synaptic plasticity. So if you recall, I mentioned that when we were looking at evoked potentials in the substantia nigra, we found that 30 Hz train, that instead of the evoked potentials being a constant amplitude, all of a sudden they started to taper out and rapidly attenuate. However, the first stimulation pulse in that train was actually larger than the baseline amplitude. And this is the most likely reason for that is that the previous stimulation train at 20 Hertz induced a synaptic potentiation. So we're interested in sort of following up on this potentiation response. And what we're able to demonstrate is that there's an increase in inhibitory synaptic plasticity that occurs after high frequency stimulation. So to demonstrate this, we delivered stimulation test pulses at 1 Hertz. We evoked our constant amplitude evoked potentials. And then we delivered a train of continuous high frequency stimulation, which is intended to essentially mimic continuous high frequency clinically applied DBS. But we only did it for 10 seconds. And then we went back to our 1 Hertz test pulses, and we found that the evoked potentials were now roughly twice as large as they were prior to delivery of electrical stimulation. But not only that, we also measured the duration of neuronal inhibition of each stimulation pulse. And after high frequency stimulation, that duration of neuronal inhibition doubled, and simultaneously as the evoked potential amplitude doubled as well. So most likely the amplitude of that evoked potential determines for how long the neurons are going to be inhibited for, and after continuous high frequency stimulation, you potentiate this response. So the evoked potentials are larger, and the neurons are inhibited for a longer amount of time. We wanted to expand on these initial findings, so we replicated the experiment using single pulses now at various pulse widths. We did this in the substantia nigra pars reticulata, the SNR, and we also did it in the GPI, the globus pallidus internus. And these two structures share synaptic inputs. So they have very similar synaptic inputs, and they're both the output structures of the basal ganglia. And they receive a large predominance of inhibitory GABAergic inputs. So in 28 patients with Parkinson's disease, we delivered those single pulse test pulses at 25, 50, 100, 150, and 250 microseconds to be altered the pulse width before and after a train of continuous high frequency stimulation. And what we found was that before high frequency stimulation, which are the dashed lines in the top graphs here, before high frequency stimulation, as we increase the stimulation pulse width, the amplitudes of the evoked potentials progressively increased, and so did the neuronal inhibition. So essentially, the hyperpolarization scaled with the amount of energy we're delivering with each pulse. So the larger the pulse was, the stronger the hyperpolarization was. But after a continuous train of high frequency stimulation, both of these responses were enhanced, and they also scaled with the pulse widths. Not only that, we found that, so we performed this stimulation paradigm, so we delivered 10 pulses at each of those pulse widths. And of course, we're recording the inherent firing rate of these neurons at baseline and throughout. So after the delivery of high frequency stimulation, and then after completion of that stimulation paradigm, which is approximately 2 to 2.5 minutes of the paradigm, the inherent baseline firing of the neurons was also persistently reduced, which is another indication of enhancement of inhibitory synaptic efficacy. So to conclude this third and final mechanism, we found that inhibitory synaptic plasticity is enhanced after continuous high frequency stimulation. This may occur as a result of a presynaptic increase in how much neurotransmitter is being released, or it may be due to a postsynaptic increase in the prevalence of receptors on the postsynaptic terminal, or an increase in the efficacy of those receptors. However, this enhancement of synaptic efficacy is not necessarily very relevant to the current mechanism of deep brain stimulation, because it's clinically deep brain stimulation is delivered continuously. It's on all the time, right? So the more likely therapeutic mechanism of deep brain stimulation is synaptic depression. However, this finding may have implications for the development of novel stimulation paradigms, which can be used to periodically enhance synaptic efficacy as opposed to continuously depress it. So to briefly summarize the three mechanisms I spoke about, electrical stimulation causes release of neurotransmitters from presynaptic terminals. That's our synaptic activation sort of hypothesis. However, if the stimulation frequency is fast enough, it doesn't allow the synapses enough time to recover, causing synaptic depression or synaptic fatigue, which is likely a very important therapeutic mechanism of deep brain stimulation. However, we found that in the SNR and in the GPI, electrical stimulation can be, or an enhancement of inhibitory synaptic plasticity occurred after a train of continuous high frequency stimulation. So essentially, depending on how it's applied, electrical stimulation can be used to excite neurons by driving sort of entrainment of neuronal activity, depending on the structure you're in. If you find a structure that has predominantly glutamatergic synapses, and you use a particular frequency that's not too high, you can entrain neuronal activity and drive neuronal activity. It seems that irrespective of the of the weighted composition of inhibitory and excitatory synapses. If you use a frequency that's high enough, that's fast enough, that depletes these readily releasable neurotransmitters, you end up depressing neuronal activity. And we also showed that we can alter or enhance the efficacy of synapses by essentially changing what effect they have, which may be sort of a future mechanism of neuromodulation technologies. Yeah. So just quickly, I want to acknowledge once again my wonderful clinical collaborators at Toronto Western Hospital, as well as at Julian, and of course my mentors and thesis supervisors, Dr. Popovich, who I'm sure you'll know very well, as well as Bill Hutchison. Thank you very much.
Thank you. Any questions?
With the synaptic plasticity that you were observing in the third thing you're talking about, were you able to see how long it lasted, or since you're mostly doing this during a surgery, you only look at the effect for like an hour or something?
So I'll just quickly repeat the question. The question was, are we able to determine the time course of the synaptic plasticity? And you also said the answer, which was no, because we do these experiments intraoperatively, so the patients, of course, they're awake, so we don't want to keep them in there much longer than we need to be. So the majority of these experiments that we do are only a couple minutes. The most exciting, the most interesting thing to know about the plasticity would be how long it lasts. But that's something that's more suitable for animal studies, and we weren't able to discern that intraoperatively because we'd have to sit there for potentially 20, 30 minutes, hours, or days for those responses to return to baseline. Yeah. So synaptic plasticity is essentially an alteration or a change in a response, but you wouldn't expect it to last forever. So eventually, after a couple hours or a couple days or weeks or something, you would expect it to return to baseline, but we don't know the time course of that. Yeah.
Thank you, Luca. Sorry. Really silly question. What was the shape of the pulses that you used for stimulation?
They're biphasic square pulses.
Biphasic. Thank you.
Robin.
I have a couple of questions, but thank you very much. That was really interesting. I think it's great that you were trying to understand the mechanisms by which DBS works. One of my questions is, did you have a chance to look at effects of deep brain stimulation more remotely? So obviously you change something somewhere in the brain and the brain trips us up and it compensates elsewhere, and it happens really fast. So I was wondering if you looked ipsilaterally or contralaterally at...
I guess the short answer would be no. But of course, there are a plethora of clinical studies that happen after. So the thing that's kind of interesting about the stuff that we do is that we do it acutely, intraoperatively, and we're able to measure very high sort of resolution activity within the brain. We only have a very brief, short period of time to do this. But of course, after these patients are implanted, there are tons and tons of studies that look at these sort of clinical and behavioral things that can last days or weeks or months. We can't do this intraoperatively. Yeah.
Okay. The other question I have is, do we know why the subthalamic nucleus is overactive in the first place?
Yes. Well, I mean, there's a sort of prevalent theory, I guess. So I saved this slide just in case. I didn't really talk too much about sort of the intricacies of connections of the basal ganglia and the basal ganglia connected to other structures of the brain, because I think it would be a little convoluted. But if anybody's interested, and of course you are. So this is sort of how the basal ganglia structures are connected in a healthy person. And essentially what gets degenerated is this projection from the substantia nigra pars compacta. So this is a structure that delivers dopamine essentially to the striatum. The striatum is sort of the input structure of the basal ganglia. And this projection sort of, let's say unhealthy, mediates two separate distinct pathways. So one pathway, which is this one, inhibits movements, that suppresses voluntary movement, and this pathway here facilitates movement. And in the absence of dopamine, this pathway becomes increased, and this pathway becomes decreased. So now we have an oversuppression of movements, which is a cardinal feature of Parkinson's disease. Might as well use this opportunity too to give my theory of how subthalamic deep brain stimulation works, which is, I mentioned that when we deliver continuous high-frequency stimulation, we're just suppressing the output of the subthalamic nucleus, which is a structure that's basically right in the middle of this overactive pathway. So if we suppress this, then we sort of bring these two pathways back to a balance.
Okay. Thank you.
Any other questions?
With back to the plasticity, you observed it with enhancing the inhibition. Did you get a chance to see if you could enhance activation in a different area, for example, or did you not have the opportunity to check that because you were only doing an experiment in one area?
Yeah, so haven't gotten there yet. That would certainly be interesting. Yeah, we haven't gotten there yet, I would say. So a lot of this is sort of what kind of patients you have access to. So most patients that we get have Parkinson's disease, we implant the SCN and the SNR, and potentially the GPI. So we get very, very, I would say much, much fewer patients who get electrodes implanted into the ventral intermediate nucleus, which is a structure that has a predominantly excitatory input. Yeah, so we haven't done that yet, but potentially in the future.
I know this isn't what you worked on, but do you think there's any potential with deep brain stimulation for enhancement of cognitive functions and
Potentially. I would say that how it's currently applied, probably, possibly not. That would be my best guess. Which is why I'm sort of interested in this enhancement of plasticity. DBS has been tried for Alzheimer's disease, and basically most applications of DBS, I think, rely on the efficacy of subthalamic DBS for Parkinson's disease. So I think a general assumption is we can put an electrode in, stimulate at a high frequency, and we expect some, good result. But I think for cognitive function, it may be beneficial to not suppress activity, but rather try and enhance it or drive it. Yeah, that's my best guess. But of course, all these things I think, require animal experiments and, making sure that the safety and efficacy of these things works. There's no long-term effect. So I think it can be tricky because you don't know what the long-term consequences of these things could be. You could enhance plasticity and it could have a good effect for a certain period of time, and then something could go wrong down the road and you may not be able to reverse things. So yeah, I think understanding the mechanisms can help us expand the application of DBS. But I think with the current technology, which is just delivered continuous high-frequency stimulation, yeah, I think we're limited.
I know Andre Lozano was starting to implant in the Papez circuit, and I think he was getting some interesting findings.
Yeah.
And that was
Yeah,
presumably
exactly.
not inhibition, it would have been excitatory.
I don't remember off the top of my head. Do you know, Bill, when these Alzheimer's patients are programmed, are they programmed at like 100?
I think they still don't know exactly the
Yeah.
right parameters, but it's in the phonics, so the
Yeah,
idea
exactly.
is it would activate the white matter tract and an antidromic activation of the outflow of hippocampus. That's the idea.
Yeah.
The other idea is maybe there's some kind of neurogenesis, but in those patients there's so much degeneration, it's hard to know that a 1% increase in neural number would be significant. Yeah, so that's sort of ongoing. There were the largest study that has just been completed with that showed that the older individuals seem to do marginally better, but the individuals that were under 50 seem to do worse. So I don't know, there's a bit of a story that you can work there, but it might be kind of regression of the mean. So I think the jury's out on that. But
Yeah.
I guess that was my comment, because you talk a lot about synaptic mechanisms here, but you haven't really used an antagonist. You haven't really proven it, right? So do the slice studies sort of support the synaptic theory you proposed predominantly?
Which theory?
Well, a lot of people have done, that all of the, say, for example, the long-term inhibition is a continuous sort of inhibitory synaptic effect. Because some people talk about inactivation of sodium channels,
Right.
and this could be part of the mechanism. But actually, to prove it's synaptic, right, you have to give like acutylene or GABA
Yeah,
antagonist.
so we can't do that intraoperatively with our human patients, but slice studies have shown that you can selectively, you can pharmacologically isolate essentially explicitly excitatory or explicitly inhibitory synapses. And I don't know if you're aware of the paper, but there's a paper from Germany that just came out that pretty much replicated some of the studies that we did here with the various stimulation frequencies, and in particular they're looking at high frequency stimulation. Because in that paper we wrote that high frequency stimulation has sort of a non-selective synaptic depression effect, and so they replicated the study in animal slices, and they pharmacologically isolated within the subthalamic nucleus only the excitatory synapses, only the inhibitory synapses, and showed that indeed high frequency stimulation depressed both excitatory postsynaptic potentials and inhibitory postsynaptic potentials. That's not to say that that's the only mechanism involved. That's sort of a local.
I was just wondering when your thalamic thing, whether that early burst is sort of, because thalamic neurons are kind of different, have a lot of recurrent collaterals, right?
Yeah.
And it would be interesting, say, to have a model that might sort of predict those effects, say, with melasma, that early burst might be sort of an antidromic activation of the cell itself.
Right.
So, because we know probably reactivating fibers, but we don't know where those fibers exactly are going in each case. So.
Yeah. A lot of intricacies left unanswered, I guess. Yeah. One thing that one of sort of my goals for going beyond this sort of more basic neuroscience research, which I am working on in Tübingen now, is sort of looking at any sort of clinical implications of enhancing plasticity and testing out novel stimulation paradigms. So that's sort of something that's of great interest to me, is following up clinically on mechanisms other than depressing the synapses. So hopefully in the near future we'll have some cool results on that front.
One more question?
Is it possible to implant a second electrode along with the permanent stimulating electrode, or like in the way that you were doing the experiments, or does the amplitude become too high once it's a permanent tube?
I mean, you can implant. Sorry, for what purpose?
To be able to get those sort of ongoing, the ongoing information, because your restrictions is that you can only do the experiment for an hour or so.
Yeah. So one thing is that I guess technically you can implant an electrode and you can leave a microelectrode in there, but there's no technology right now that can enable you to continuously record from that microelectrode, for example. One thing that is a big area of research and an area of interest in these patients. So first of all, I should qualify that the microelectrode is very small. It doesn't get left in the brain.
Yeah.
The DBS macroelectrode gets in there. The macroelectrode can't record action potential firing, but it can record local field potential activity, which is essentially sort of a summation of probably millions of neurons firing simultaneously. So people look at neuronal oscillations essentially, which are manifestations of dozens or thousands or millions potentially of neurons. So people can record from those chronically. You don't get the same sort of information as you would from single neuron recordings, but there are lots of studies that look at that. With that, the DBS electrodes have generally four contacts. So you can put at least 2, most likely 3 contacts in the SCN. So technically, yes, you can record local field potential activity with one and stimulate with the other. The problem is, as far as I know, you can't record and stimulate at the same time. So when the stimulation turns on, it shunts recording capabilities. But you can look at things immediately after stimulation, for example. Yeah.
Is that because the microelectrodes would move around too much once people are living? Be able to
So this is more of a hardware thing?
keep it- Yeah.
Yeah, so when you deliver stimulation, I guess maybe some of the newer ones can do both at the same time, but DBS artifacts are really large when you use a big contacts.
Yeah.
The other thing is if we left a microelectrode in there and tried to record from persistently one neuron, it would most certainly move around and we'd lose that. Yeah, so there are some ways of sort of attempting these kind of studies, but they all have their. Actually, to answer your question, I think a lot of the times when you stimulate, you can't record at the same time because there's sort of one stimulator, one amplifier. We benefit here from having 2 separate amplifiers and 2 separate stimulators. So when you turn stimulation on here with the DBS electrode, the recording capabilities get shunted essentially. So we can't get sort of the information between pulses and stuff like that.
Thank you, it's great.